A warm good morning to all my dear students. Today we are going to discuss yet another important topic, that is biotechnology and its application. We will be specially considering the plant biotechnology. As you can see, plant biotechnology is used for genetically modified crops, for transgenic plants, for plant tissue culture, for therapeutic plants, as well as for molecular farming. Molecular farming is nothing but production of therapeutic products in using plants as a experimental organism. Genes that are of human origin are introduced into the plants and they are produced in higher quantities so that they can be marketed. It is a very widely used tool these days and is being used for producing more and more quantities of certain drugs and antibiotics, antibodies, etc. by the pharmaceutical company. One of the examples is the human serum albumin, which was produced in 1900 in tobacco and potato plants. Going into the next slide are the plantibodies. Plantibodies are the antibodies which are genetically modified and they are produced in the plants. One of the world's first clinically tested plantibody was CaroRx. But now the question is why to choose plants where we can choose animals, microorganisms, etc. Have you ever thought? Yes, the answer is the plants are very much flexible in production and they are low cost. And most importantly, they have no ethical issues. That's why we are choosing plants for all the research purposes as well as for producing all the therapeutic drugs. There is a slide which is showing tobacco. I can show you that tobacco, it has been used for a number of drugs. You can see for production of IgG against nematode, IgG for Streptococcus mutans, colon cancer, rabies. So tobacco, you can see it has been used for producing a number of antibodies and the specific purposes are also being highlighted. Please go to the slides and try to remember these slides as it is very much important for your MCQs as well as for other competitive exams. Here is a slide which is showing tobacco and sunflower. They are being used in production of growth hormones, and here are the certain references and the references and the reference books and the reference PDFs from where this species and their recombinant human products has been taken, and you can see that tobacco is used for production of growth hormone, serum albumin and many other recombinant human products. These are certain proteins that are used. Then going into the next slide is the Bacillus thuringiensis or the BT, which is a soil bacteria having the insecticidal properties. This Bacillus thuringiensis has been used in certain plants for making the plants insect resistance, for developing varieties which are insect resistance. This is a very much innovative technique in biotechnology, and you can see the whole mechanism. I have shown it in the form of a flow chart here, and you can see that BT has been sprayed to the crops. They are when eaten by the plants, they are at first they are in a inactive protoxin forms. You can see I have highlighted the word here, inactive protoxins, and as soon as it gets the gut of the insects, it gets converted into an active form. And as it reaches the active form, the guts of the insect swells and it leads to the death of the plant. This technique has been used in production of numerous commercially available crops. You can see BT cotton, BT rice, BT brinjal, which is a very much controversial topic these days, and we will be having a discussion today on this BT brinjal, and it has been commercially available crops. Then these are certain other examples, that is the flavor saver tomato and golden rice, and you can see that flavor saver tomato, it has been found that they have a longer shelf life, and the golden rice, they are very much rich in beta carotene. These are also certain biotechnologically developed genetically modified organisms. Another technique that is used is the Agrobacterium tumefaciens mediated gene transfer, which is also used to produce genetically modified crops. In order to make the crops disease resistant, the soil bacterium is used, and this soil bacterium is known to cause the crown gall tumor in the plants. and these are also used in the production of the disease resistant plants. Then the plant tissue culture is also another adventure and another application of plant biotechnology, where a number of plantlets can be produced from the basic plant cells, from the leaves, stems, roots, any kind of the plant. It can be used as an explant and produce a number of plantlets which can be generated for higher production, or it can be used for the mass culture of certain plants. This is the layout showing the technique of plant tissue culture. And in the next class, we will be discussing about the future of genetic engineering. Here is an image which is an extremely improbable image, but there is a possibility that in near future, we might develop certain technology that we can have our dear ones cloned. And even after death, we can become immortal. We can have our clones and we can be immortal. And this is the future of genetic engineering. In the next class, we will be discussing on the nanobots, nanotechnology, and the future of genetic engineering. Thank you.